Okay. Yeah, so today's class, starting with the gastrointestinal physiology, will be kind of an overview of all the secretions that happen throughout as chyme and food moves through the GI tract. All the different secretions, everything that's contained in that, a little bit of the digestion process, and then we'll probably spend more time tomorrow talking about the specifics of digestion of each macronutrient and absorption of those as well. So yeah, so you got a little bit of an overview in the pre-class video of all the different hormones released in the GI tract, right? It's a lot. Continuing on with our fun from the endocrine system, lots of those hormones, and then yeah, and then just a bit of the kind of functional anatomy as well. Have you guys gotten into the abdomen yet in the with the donors? Yeah, okay. Yeah. So yeah, but just a little bit of the functional anatomy there you got. So let's talk about a couple unique things here. So again, we have another portal system in the GI tract, okay? Just like we had a portal system up in the brain in the hypothalamic pituitary portal system. This one is definitely a more well-known portal system, the hepatic portal system. Right? And so the idea of a portal system is that we have two capillary beds before return to the heart. Right, so typically we've just got the one capillary bed. We go on the arterial side, capillary, venous side, back to the heart. Right, with the portal system, again, we've got arterial side, capillary bed, vein, another capillary bed, venous system, back to the heart. Okay, so that means that second capillary bed typically will have a unique function, right? It doesn't typically have the same function of delivering oxygen. It has a unique function, right? So the first capillary bed, which is going to be found in the small intestine. Okay, so we can kind of see this image of it here. Okay, so small intestine needs to absorb products. So first of all, that first capillary bed is going to deliver oxygen to the small intestine. All right, but then, so it's kind of function one, but then it also absorbs nutrients. Right, so we kind of have those two functions there. We do have oxygenated blood coming there, so it's going to be delivering that oxygen and the nutrients for the cells there to use, right? But then it's also going to be absorbing all the nutrients that are absorbed or digested first and then absorbed in the small intestine. Okay, and then we get that. Oops, we get to that second capillary bed, which would be more in the liver, right? So the liver, gosh, sorry, the liver capillary bed. Is going to be just to drop off nutrients. So most of the nutrients that are absorbed in the GI tract need to be processed in some way by the liver. Okay, all the carbohydrates can kind of have a quick stop in the livers. A lot of the glucose can be turned into glycogen at that point, right, and stored in the liver. We'll talk about how fats are absorbed. They're not absorbed directly in this capillary bed, but they end up back in the bloodstream via the lymphatic system, and then they are brought to the liver. And we'll talk about that process tomorrow, probably, how kind of turned into the normal cholesterols that we look at. And then with proteins, we're going to be breaking them down into amino acids, and those are going to be taking a stop in the liver. Some of those will stay there to be used to create protein. Some of those will just continue on in the bloodstream. But yeah. Andy?
System, is there just one vessel that connects the two kind of portals, I guess you could say, in the liver and the small intestine, or is it a series?
It's a series because you've got it's not really just the small intestine. That's kind of what I'm focusing on because that's where most of the nutrients are absorbed. But it is also from yeah, from other parts of the intestine as well. It's all going to come back to liver. But then there at some point you do get to the hepatic portal vein, which is the big one that's going to take that all of those are going to dump into from the GI tract. And then it goes, then it's going to break up into smaller ones to spread it throughout the liver. And then there'll be obviously lots of capillary beds throughout the liver. But yeah, so at one point there is just that one big vessel, the hepatic portal vein, but there's a lot that feed into that. And then that branches out a lot after that. But yeah, good question. Yeah. Yeah, so that's the hepatic portal system. One of the things we already talked about in terms of kind of pathology related to this would be in type 2 diabetes. We have one of the biggest risk factors of type 2 diabetes is the level of visceral fat that a patient has, right? So we've talked about subcutaneous fat, which is just the fat underneath our skin, right? But visceral fat is that fat that's built up around our organs and underneath our abdominal wall, right? Underneath all the muscle, right? So it can build up around our heart, in our heart, around our liver, other places. But there's a big depot of visceral fat right next to that hepatic portal vein, right? And again, that visceral fat is highly metabolic, meaning that it's constantly breaking down the fat, much more so than subcutaneous adipose tissue. So it constantly releases free fatty acids into the blood. And since that one is right there at the portal vein, the next stop is the liver. The people that have a lot of visceral fat end up dumping a lot of free fatty acids into their liver constantly, which eventually leads to some fat buildup in the liver, which eventually is a risk factor for type 2 diabetes, right? Because one of those primary pathologies in type 2 diabetes, which was one of the essay questions, right, is the fact that your liver becomes insulin resistant, right? Your liver becomes insulin resistant, and the insulin's supposed to tell the liver when there's plenty of glucose in the blood, right? Um, but when that mechanism isn't working properly, there's plenty of glucose in the blood, insulin's released, supposed to tell the liver to stop releasing glucose, but it doesn't, right? It does that mechanism is broken, so the liver keeps dumping glucose into the blood, and that's one of the three reasons why type 2 diabetics have high blood sugars because of their liver, right? And so a lot of that kind of stems back to the visceral fat that's built up around the portal vein, which ends up dumping a lot of fat into the liver and causing some what used to be called non-alcoholic fatty liver disease, right? So, okay, questions about any of that? Okay. All right. Well, let's start at the top of the GI tract and kind of work our way down. We'll talk about each kind of location and then what secretions are there, how much of each. If you want to keep track, it's kind of fun, kind of have like a little scorecard. You can go to slide 11, this one, and then we're going to add everything up from each spot. So you can do that for fun. Sure, you'll have a real fun time with it. But yeah, so let's start in the mouth. Okay, so the salivary glands um, just are continually releasing saliva, right? And a lot. Okay, we're talking about 1 to 1.5 liters per day. Okay, so it's a lot of fluid coming from these little glands in our mouth, right? Or around our mouth that release into our mouth. Okay. Um, I mean, think about it, right? We're talking about like a picture like a 2 liter soda bottle, right? Like almost that, right? 3/4 of that every day coming into our mouth, right? Most of that ends up going down our GI tract, right? And so the majority of that is going to be due to when we have parasympathetic activation of these glands. Okay. I guess the other kind of fun stat here is that these guys secrete their own weight every minute. They're not very big, right? But every minute that they're secreting things, it's the equivalent of the weight of the gland, right? And so it's just a lot of fluid that they're producing and pumping out constantly, right? So the parasympathetic innervation includes uh, two different cranial nerves, right? The facial nerve and the glossopharyngeal nerve, 7 and 9. Um, and there's a couple different reasons why, a couple different stimuli for this. Okay, so one is presence of something in the mouth. So we actually have touch receptors, pressure receptors in our mouths, right? And that those pressure receptors, when they're activated, causes release of these fluids. Okay, so that this is all shown over here, right? So we've got pressure in the mouth leads to salivation, right? So we've got these two different glands, the submandibular, sublingual, right? So we're seeing that release there. And then also things that are like CNS stimuli, right? Brain stimuli. We see food, we smell food, the taste, right? Even the certain sounds can make us salivate, right? So all these different things kick in there as well. So those are the two primary stimuli. Okay. And so then the question is, what's in all this fluid, right? So amylase is one. So amylase is an enzyme that breaks down starch. we can just say, sorry, let's try that again. We'll start, there we go. We can say polysaccharides. Right? So complex carbohydrates, right? That's what these break down. The kind of cool little science experiment you can do on yourself is take like a saltine cracker or like a piece of white bread and stick it in your mouth and just let it sit there for a while. It doesn't taste sweet at first, right? But the longer you leave it in there, it starts getting sweet. And the reason for that is because polysaccharides don't activate our sweet receptors, but disaccharides do, right? And so when we start breaking those polysaccharides into some disaccharides, then we start tasting sugar, right? So that you can try that later if you want, but yeah, so that's one of the key things that's digestive or that's chemically digested in our mouth is carbohydrates. Really, the only thing that's chemically digested in our mouth is carbohydrates. Okay, and then we've got some things to help us with different yeah, bacteria that we may be ingesting. All right, so we have some immunoglobulins there. We have some lysozymes there that can help us kind of break through membranes of bacteria to destroy them. So we're not ingesting that, right? And then we have mucins, right? So mucins are kind of the precursor to mucus. And so when mucins hit the saliva, get into the mouth and kind of fully liquefy, then it turns into mucus, right? And that mucus helps to lubricate everything as it's going down the GI tract. And then we have some bicarbonate too. And so typically, in general, foods tend to be a little bit more acidic than they are basic. Um, and this helps with the function of that amylase enzyme that it needs to stay in a little bit more of a basic environment for it to work. So yeah, so those are the primary components of what's released in salivary glands. And then obviously water too. There's lots of water as well. But yeah. Questions on that? Okay, so now we're making our way down through the esophagus, right? There's not much actively going on in the esophagus. It's just kind of a transport thing, right? There's mucous membrane there that's helping things stay lubricated and smooth muscle to push everything down. We'll talk about smooth muscle contractions next week and kind of how all of that works. But so that's why we're kind of skipping that part and going down to the stomach now. Right, so the stomach secretes lots of fluid as well. Okay, so we can see here per day, about 2.5 liters of secretions by the stomach. Okay, and this was this little structure here from a gastric, what's called a gastric pit. All right, so these little kind of what's called an invagination inside the lining of the stomach has all kinds of cells, all kinds of specialized cells that release different things. And so this is where all the secretions are coming from. Okay. And so, yeah, so for each of these, we'll kind of talk about these two questions. What do they secrete and what are the functions of those things? Okay. So the first one to talk about is the parietal cells. The parietal cells release hydrochloric acid. Okay. So very strong acid. And the purpose of releasing hydrochloric acid is to drop the pH, obviously. Okay. And this is very helpful for protein digestion. Okay, that low pH denatures proteins and helps those proteins kind of start falling apart, which then we'll be able to use some enzymes to do the actual breaking apart, but it helps to kind of unravel them first, right? And that's what that hydrochloric acid does. And then also going to activate pepsinogen. Okay, we'll talk about its role in a second, but that low pH is important for activating pepsinogen. Okay, and then we've also got something else released there called intrinsic factor. Which I think, yeah, I think that I already mentioned that one in the pre-class video, right, with absorption of vitamin B12, is that what it was? Yeah, thank you. Yeah, I wasn't quite sure. Yeah, yeah. So intrinsic factor is important for the absorption of vitamin B12, but not until the end part of the small intestine. But the factor that's necessary for it is released way up here in the stomach. So we'll just say B12 absorption, but not right here. Okay, sorry, that was too many things to write in that little spot. Any questions on what I wrote there? Okay. All right. So those are the parietal cells. Okay. So we can see those guys here. They have a unique look to them. And they have lots of mitochondria because it takes a lot of active transport to push to push all that hydrogen out when there's already a lot out there. Okay. And we'll talk about that just on the next slide. Okay. So that's the first one. Chief cells are next. So chief cells release pepsinogen. Okay, and when that pepsinogen gets converted by the low pH or gets activated by the low pH, then this helps us with protein digestion as well. Okay, so the low pH helps to kind of unravel the protein, right? But then the pepsinogen is actually enzymatically cleaving some of those bonds that hold together the primary level of the protein. Okay, so these ECL cells are called enterochromaffin-like cells. So we see those here. Okay, we'll talk about enterochromaffin cells in a minute, but enterochromaffin-like cells, they kind of look like them, but they're not them. That's why they're named that. Okay, so these ECL cells, they release histamine. Okay, and the histamine is one of the primary stimuli for the parietal cells. So they stimulate the parietal cells to release hydrochloric acid. Yeah, go ahead.
Cells are dropping pH. Are they doing this through pepsinogen or are they doing this through something else?
The chief cells are not dropping the pH. The parietal cells are dropping the pH through the release of hydrochloric acid. But one of the functions of the low pH is to activate pepsinogen. Maybe that's where the crossover happened. But
It's by how they both are involved in digestion. So for
Yeah.
parietal cells, it's through the drop of pH. But what would it be for chief cells?
Yeah, for chief cells, it's the fact that they release a proteolytic enzyme, pepsinogen, right? But it needs to be activated by what's released from the parietal cells, right? So there's kind of two different parts of protein digestion that happen in the stomach. One is the low pH that happens because of the hydrochloric acid release from the parietal cells. And so that denatures the protein a bit, right? And kind of like that makes it so there's a lot more amino acid, a lot more places where enzymes can cleave, right? Because it kind of opens everything up. And then we've got pepsinogen released from the chief cells. When that interacts with the low pH from this, that activates it, and then we can enzymatically start cleaving some of those amino acid bonds, some of those peptide bonds that hold the protein together. So yeah, good question. Okay, other questions so far? Yeah, Sam?
So with protein digestion with parietal cells and the chief cells,
Mm-hmm.
the parietal cells essentially are like breaking down the more complex folding, and then the chief cells, like the enzymatic part, that's where you see the amino acids getting chopped up individually. Is that right?
Yeah, we're not going to get it to individual amino acids yet. That'll happen in the small intestine, but this is kind of cleaving really long polypeptides into a little bit less long polypeptides. So yeah, we'll see with some enzymes released from the pancreas and from the small intestine, from the pancreas, all the different pancreatic enzymes and the brush border enzymes that we see, those would be the ones that finally cleave it into like individual amino acids. But right now we're just kind of chopping up long chains into smaller chains. Yeah. Other questions on that? Yeah. And
Pepsinogen the inactive form of pepsin.
Exactly. Yeah. So pepsinogen is the inactive form of pepsin. Yeah. Yeah. Okay. Good questions. Other questions? Okay. All right, so then we've got what are called mucus neck cells, okay, and we can see those right here. So that's kind of at the top part of that intestinal pit or the the pit, the gastric pit. Um, and so these are the stem cells that are making more cells that are going to help to line the GI, the stomach lining. Okay. This is the type, the thing that lines our GI tract is epithelial tissue, right? One of the characteristics of epithelial tissue is it's highly proliferative, meaning that the cells get damaged all the time, right? And so we need to replace them all the time too, right? And so we need to have a big group of stem cells ready to go so we can constantly replace those cells. And that's what those mucus neck cells are. So um, yeah. Okay, and then the ones on the surface, those would just be these guys here. These are the ones that are doing they already are making the lining, right? So they're secreting mucus. All right, and they're doing the protection of that lining. They're trying to seal off the stomach from the rest of the body, right? That's what they're doing. Okay, so mucus secretion, protection. Okay. And then lastly, we've got the G cells, which are going to release gastrin. So gastrin is going to be an important hormone we'll talk about. And these G cells are found mostly only in the antrum, which is kind of more the bottom part of the stomach there. Right, so we've got the fundus is the top part, the body is the middle part, and then the antrum is the bottom part of the stomach there. We can see those different locations. So the G cells are found mostly down here. And a lot of the times they try to make it easy and make the primary hormone that one of these enteroendocrine cells release the same first letter as the cell that it releases. So G cells and gastrin. One way that anatomists try to make it easier on you guys, but it's not always that way. But yeah, go ahead, Alexis.
Stem cell,
Yeah, so the stem cell, so the lining of the stomach is pretty much under, chemical assault all the time, right? Because the lining, our stomach, the pH typically sits around 1.5 to 1, right? And so that's highly damaging to cells, right? And so first of all, we need lots of mucus release there to kind of buffer that acid from destroying the cells directly, right? And it's not only chemical assault that the stomach is under, but physical assault too, because the stomach has 3 different layers of muscle and it can just smash around in so many different ways, right? So between the physical pressure of the stomach smashing itself and then the chemical pressure of that low pH, there's a lot of damage to those cells all the time, right? So part of the job of those surface cells is to make the mucus to protect themselves. That's what this yellow, lining is representing as a mucus, right? So that's the mucus cells is providing that protection. And the last thing we'd want to do is to poke a hole in our stomach and let that acid get out into our abdominal cavity, right? So the primary role of these surface cells is to make the mucus to protect the lining and just to be a physical barrier in the stomach. And since they're damaged so often, we have to have cells right below them that are constantly replacing those damaged cells, right? And that's what the neck cells are for. So mitotically dividing cells that are just constantly replicating and sending new cells up to replace those damaged cells in the lining. Yeah, thank you. Okay, other questions on that? Okay, so a little bit of the control of this, right? We talked about there's all these different things secreted, talked about a couple stimuli, but let's talk about the full picture there of stimuli of these gastric secretions. Okay, so the 3 primary stimuli, number 1 would be gastrin release. Okay, so this is a hormone, right? All throughout the GI tract, we have these individual cells that act as little endocrine glands, right? We typically think of endocrine glands as like a full organ, right? But we can also have what's known as the diffuse endocrine system, right? Which is individual cells all throughout the body releasing things into the blood, releasing hormones into the blood, right? And so we have that all through the GI tract, and we call this the or we call them in general enteroendocrine cells, right? So entero having to do with the GI tract, endocrine obviously releasing things into the blood. So those G cells don't release into the stomach, right? They release the other direction, right? When we look at these cells here, right? Does it have the G cells on here? Maybe not, huh? Yeah, I guess I don't see them there, but instead of releasing this way into the pit so it can go out into where the food is, it's going to release back this way, right? Into the blood, right? So that's its endocrine function, okay? So releases gastrin, goes into the bloodstream, and we can see right here, this is a small version of one of those little pits, right? We see the G cell releasing this way, it goes through a capillary, and it's going to interact with some adjacent, an adjacent gastric pit, right? That's what this is. And it's going to activate. The gastrin does a lot of things. We can skip to this one. It activates the parietal cells to release hydrochloric acid, activates chief cells, and activates those ECL cells. So all of those are kind of controlled by gastrin release. Okay. Histamine is also one of the stimuli, right? So we can see histamine here being released by those ECL cells. Histamine is going to go up to that chief cell and cause it to release. Okay, and then also we have nervous system control here as well, right? So we've got neurons coming from right here. You can see them right here. So these would be some enteric nervous system neurons interacting directly with those ECL cells as well. So we can see, acetylcholine being released here, causing some more histamine release. That would be part of it, but then also enteric nerve ending here, going straight to the chief cell itself, more acetylcholine release, causing it to release its secretion. Okay, so those chief cells, again, are the ones releasing pepsinogen, right? That's what the P stands for. So we see the little P being released there. Okay, so that's the chief cells. But then we can see that also gastrin goes directly to the parietal cell, and histamine goes directly to the parietal cell too, right? And we can see the neuron hitting here, right? So all of them, all these things are interacting directly with the chief cells to release pepsinogen and directly with the parietal cells to release hydrochloric acid. Okay, so it's kind of a complicated little figure from your book, but it does a pretty good job in kind of laying all that out. You just have to spend some time with it. Yeah, Chesna? Gastrin release. Yeah, so the gastrin release is going to be stimulated by the because it does that not actually show that on there? Yeah, so the two things it's showing here are number one, the presence of broken down protein. Okay, so that's our primary stimuli for gastrin release. And then we also have some nervous system control here. And that GRP is gastrin releasing peptide, or gastric releasing peptide is just the neurotransmitter release there by those neurons. And so those would be the two primary things that cause gastrin release. And we'll have a slide in a little bit where we'll kind of summarize the hormones, and we'll look at the stimuli for the release of each, but the image does show the two primary ones there. So when food gets into the stomach that contains some proteins, that kicks off this whole process for the most part. Yeah, good question. Yeah. Okay. Other questions there? Okay, so then let's just take a quick peek at the parietal cells. Okay, so the parietal cells are unique cells, right? They look unique, and they have lots and lots of mitochondria. Okay, so they need to produce a lot of ATP. Okay, they have 3 different receptor types here. Okay, we just looked at 3 different stimuli to cause them to be released, right? So then all the receptor types have been identified for those stimuli. Okay, so we can see a muscarinic receptor right here. That's what the M3 stands for, right? Acetylcholine is binding to that. Okay, we can see an H2 receptor, which is the histamine one. Okay, and then we can see this other one. It's not a receptor type we've necessarily talked about before, but CCKB, but that's the one the gastrin binds to. You don't need to, I wouldn't quiz you on the receptor names, but it's just showing the 3 receptors and those different ones there. Okay. So the big thing that all of these activate is activating the release of hydrochloric acid, right? And so pushing that acid out, right? And so lots of active transport. Right. And as you guys know, active transport requires ATP, right? Mitochondria is where the ATP is made, right? So we got lots of mitochondria here. These cells use lots of energy to push hydrogen ions into a space where there's already a lot of hydrogen ions. Okay. Yeah, one of the stats I have written down in my notes is that um, these parietal cells can still push more hydrogen ions into extracellularly when that concentration gradient is over 1 million fold. So meaning there's already 1 million fold more hydrogen ions outside than there are inside. That's about when they finally kind of tap out in their ability to push more hydrogen ions in, right? So there's no other space in the body that the pH can get anywhere close to this, right? Down to 1 is super, super low, right? So these are very unique cells to be able to do this. Okay. Questions on that? Okay. All right. So moving on now to kind of this big section of the GI tract with these associated organs, right? So the duodenum is the first part of the small intestine, right? The shortest part of the small intestine. It's the part that connects to the stomach, but the pancreas is right there, kind of hugged by it, right? The duodenum kind of wraps itself around the pancreas a little bit. The liver is right there. The gallbladder is right there. So all of these kind of accessory organs or the organs of the GI tract that don't include the tube itself, they're all right there around this area. Okay, so the small intestine itself adds about 1 liter of fluid per day to this, to the total secretions. A couple of things I'll point out is we've got these different ducts here. Okay, so there's this the main pancreatic duct here. So we'll talk about all the different cells here in a second, all the Acini cells that produce all the different enzymes for digestion. They're all going to dump their contents into this main pancreatic duct, which goes down, and then there's a little sphincter right here, right? The sphincter has to open up, and then all that stuff can be released into the small intestine. And then we see, we'll talk about the hepatic cells, but hepatocytes, but they make bile for us, okay? And that bile drains into all these different bile ducts, which eventually goes into this kind of big common bile duct, common hepatic duct, and those go right about to the same spot. And there's a special sphincter there called the sphincter of Oddi. We'll talk about that in a second. That has to open up to allow bile to be secreted. Okay, so all of that just gets dumped right into the small intestine right there. Okay. All right, so let's talk about what's secreted from the pancreas, okay? So again, the pancreas has both endocrine and exocrine functions, right? We talked about the endocrine functions last week, right? Talked about the glucagon and the insulin release, right? That's released into the blood. This is exocrine releasing this into the GI tract, okay? And so again, it's those Acini cells, okay? They secrete pancreatic juice into those ducts that are going to take us down to the duodenum like we just looked at, okay? This includes the juice from the pancreas itself, adds about 1.5 liters of fluid. Okay, this is bicarbonate rich fluid. Okay, so pretty high pH. This needs to counteract that really low pH that's coming from the stomach. Okay, because all the enzymes that are released from the pancreas wouldn't work under the conditions of low pH that we have in the stomach. Bless you. And so we're needing to raise that pH up a bit for the enzymes to work. Okay, and then what else is in there? Lots and lots of enzymes. Okay, most of these enzymes are in their inactive form when they're kind of stored in the pancreas. But then when they're released and they get into the right pH and the right fluid, then they get activated. Okay, and so this has a protective function because the enzymes could destroy the pancreas itself if they were activated. Okay, this is one example of this is one cause of acute pancreatitis is when phospholipase A2, which is one of our key enzymes that breaks down phospholipids that we eat, that can get prematurely activated, and it just destroys the pancreas. Right? It just inflames the whole pancreas because phospholipase A2 breaks down cell membranes, right? And so if the active form kind of gets let loose in the organ itself, then we're in trouble, right? And then it starts just breaking down all of that tissue. So all of these enzymes, for the most part, that are stored in the pancreas are not activated until they get out into the small intestine. Okay. So the primary stimuli for release of the pancreatic juice includes two things, two different hormones. One is secretin, which is going to directly activate or directly cause the release of the bicarbonate-rich juice. Okay, so it's kind of a separate set of cells that do that. And then CCK, cholecystokinin, is what is causing the release of the enzymes themselves. So we kind of need both of these things to activate the full release of the pancreatic juice. Okay. All right, questions on that so far? Okay, so that's the pancreas itself, and all the stuff come from the pancreas. But then we've got the bile too. So the bile is made by the liver cells, right? Some of it's stored in the gallbladder. Some of it isn't, right? It can just go through the ducts as well. It doesn't have to be stored in the gallbladder, okay? But we make somewhere around a half a liter per day of this. And which macronutrient is the one that bile helps us break down? Talk to each other real quick. I've been talking for a while. Talk to each other. Which macronutrient? Maybe just ask each other, what are the three macronutrients first? All right, what are our three macronutrients? Carbohydrates, fats, and proteins. Okay. Which one does bio help us with? Fat, for sure. Yeah. So fat, this is helping us break down fat. Okay. Or lipids, right? So yeah, so that's the key role of bile is digestion and absorption of lipids, okay? But then also bile helps us to get rid of stuff that we need to get rid of, okay? So cholesterol is one of those things. So we put cholesterol into our bile because it's part of the formation of bile. But then actually this is why high fiber diets lower our cholesterol levels, right? Is because when we have a high fiber diet, we excrete more of the bile, right? And in that bile includes cholesterol, right? And so then when we excrete more of it, or we defecate more of it, then we need to take cholesterol from our blood to make more bile, right? And so that lowers our blood cholesterol levels. And so, yeah, and then there's other lipid waste products that are part of the bile that get released this way too. Okay, so those are the kind of two primary reasons for bile. Okay, and what's bile made up of? So the bile acids are, there's two key bile acids, cholic acid and chenodeoxycholic acids. Okay, these are unique molecules because they're amphipathic. Okay, so amphipathic means they're like amphibians, right? They're not just polar. Polar would mean there's like positive on one side, negative on the other side. They're not just that, but one side's polar and one side's not polar, right? So that's another level of that. So one side's polar, meaning one side has a positive and negative to it, and then the other side is non-polar, doesn't have that positive and negative to it. Okay, and they form what are called micelles. Okay, so micelles are all of these guys, and there's all different kinds of shapes to them. Okay, and so in general, they actually make pretty complicated forms, but the easiest way to think about it, I think, is this image here, where we have a hydrophilic side of the molecule up here, which would be the polar side, right? That side likes to be next to water, okay? And then we have the hydrophobic side, or the nonpolar side. That part doesn't like to be next to water. It likes to be next to lipids, right? So what bile does is it goes in where there's a big chunk of lipids that we've ingested and surrounds that, right? And so all the hydrophobic side is towards the lipid. Hydrophilic side is out towards the water in our digestive tract, right? And this kind of acts like a detergent. right? How many people have been baking and the recipe says grease the pan with some butter? How many people have done that? Right? And you get some butter, you throw it in there, and you like rub it around. Feels kind of fun. But then you can't really get that butter off your fingers unless you use what? What do you have to use to get it off? Soap. What kind of soap though? Hand soap doesn't typically work, right? You got to use dish soap. Yeah, you got to use dish soap because dish soap has a strong detergent in it. And what that detergent does is it does just this. It surrounds the lipid droplets, right, and breaks them apart from each other, right? That's why it's all stuck to your fingers, is because all the lipids are kind of gathering around each other, and there's nothing to get between all the little lipid droplets, right? But that's what detergent does, is it wraps around the lipids and breaks them up into smaller droplets so it can kind of get separated and come off of your skin, right? That's exactly what bile does, is it takes these big lipid globules and kind of chunks it up into smaller lipid globules by getting between the lipids. Okay, so that's what forms these micelles. So all the lipids are on the inside of these kind of protective barriers, right? And then we've also got bile pigments. So this is the stuff that is formed when we break down our red blood cells. Okay, we recycle our red blood cells. Kind of the byproduct of that is of the broken down hemoglobin is bilirubin and biliverdin. And this is what makes bile kind of yellow. Okay, and then we've also got some alkaline juice included in the bile as well, which is good for the pH, for the enzymes to work. Right, so we need to raise that pH back up, just like the pancreatic juice did, because we're neutralizing those stomach acids, right? Okay. All right. So that's kind of it for the secretions. Questions on this so far? Yeah, go ahead, Deja. Which one? The last one? Neutralize stomach acids, that part? Yeah, cool. Thank you. Okay, let's take a little break. Let's take about a 5-minute break. We'll come back together. When we come back together, you guys are just going to be kind of summarizing this stuff from the pre-class video. So take a 5-minute break, get a drink of water, go to the bathroom, come back, and then we'll kind of just hop into this summary portion of the class. Okay, go ahead. Yes. Yeah, so CCK, I cells, again, broken down proteins and fatty acids there, okay? GIP is from the K cells, and that is going to be when we have glucose present and fat present in the small intestine, we'll get GIP release. Okay. Yeah, go ahead, Kyle.
Would it be important to know like where the cells are coming from too, or no? Like all the T cells are coming from.
Yeah, the antrum part not too important, but definitely like from the organ, right? Like whether it's the stomach or the small intestine or the pancreas. Yeah. Yeah, so then we've got yeah, VIP. So this is a neurotransmitter as well, but it does show up in the blood as well. And so again, presence of acid and fat. Okay. Somatostatin has kind of two different names really, right? What the other name for somatostatin is GHIH, right? Or growth hormone inhibiting hormone. So those terms can be used interchangeably. We can call it SS, right? For somatostatin. We call it GHIH. Either one is the same chemical structure. Okay. So it is released from the D cells, both in the pancreas and the intestines. Okay. And so if it ever just says like intestines, that means we probably have it in multiple places, have those cells multiple places throughout the GI tract. Again, acid in the intestines. Okay, peptide YY. The jejunum, okay, so that's the second part of the small intestine, right? The duodenum, jejunum, ileum, right? So the jejunum is the second part. When fat arrives in that part of our small intestine, peptide YY is released. Yeah, okay. No, not on these ones. Yeah. Yeah. So if they don't have a specific cell name on here, then it's, you might be able to find one. Sometimes when you Google it, sometimes they're just known as like enteroendocrine cells, in general. Ghrelin is released from the stomach, and it is one of our key hunger hormones. And so it's released between meals when there's not food in the stomach, right? So that's the stimulus here, is kind of lack of food in the stomach. Um, and then lastly, motilin, Mo cells, and this is stimulated by fat and fatty acids, or sorry, just fats and acids in the presence of those will cause the release of motilin. We'll talk more about motilin next week, and some of its how it causes contractions and when it causes contractions. Okay, zoom out a little bit so all of that's on the video. Questions about any of those? You still need a sec? Just raise your hand if you need another sec writing it down. Okay. All right, so then let's talk about yeah, this one here. So we've got which ones are released kind of in which part of the GI tract. Okay, so the stomach is going to release gastrin, ghrelin, and GHIH or somatostatin. Right. Small intestine, there's a lot, right? So I had to cram a bunch in there. All right, so CCK, secretin, GIP, peptide YY, motilin, VIP, GHIH. So this is where the vast majority of the business of the GI tract happens, is in the small intestine, right? So lots and lots of hormones released here. We'll talk about all the digestion and absorption next time, that happens mostly in the duodenum, but throughout the small intestine. Okay. So got those. Which one causes gastric secretions? Okay. Gastrin is our primary driver of that. Okay, pancreatic secretions. Again, we had secretin stimulating the bicarbonate-rich juice, and then CCK stimulating the enzyme rich release from the pancreas. Okay, zoom in down here a little bit. Which one inhibits secretions? Which ones inhibit secretions? Okay, as far as gastric secretions go, secretin and peptide YY are the primary ones that inhibit gastric secretions. Okay. And then somatostatin or GHIH inhibits gastric secretions as well, but also small intestine secretions. That's what that last one is. And the way to think about these, especially the ones that are inhibiting gastric secretions, is that these are all released when food particles enter the small intestine, right? So when food particles enter the small intestine, the small intestine has some work to do, right, before it can take on more, right? So it needs to slow down the stuff coming from up above so it can deal with the stuff that it just got in the small intestine, right? And so that's the whole idea. Things land in the small intestine releases stuff, and with its paracrine function tells the stomach to stop pushing so much stuff down, right? We'll see that with the sphincter being contracted too, that pyloric sphincter being contracted by secretin as well, right? That makes it so stuff doesn't keep coming down. The small intestine has to deal with what it has first. Okay. Oops, sorry. And then motilin, right, is a strong stimulator of contraction of just GI tract muscle all throughout. Okay, and then when it comes to those sphincters, CCK causes a relaxation of the sphincter of Oddi, which is the one that allows for bile to be released into the small intestine. Okay, and then yeah, VIP is in general just leading to sphincter relaxation kind of throughout whichever neurons it's, we're activating and releasing it in those specific spots. And then secretin is causing the pyloric contraction, which again, that's the sphincter allowing stuff to come from the stomach to the small intestine. So secretin going to go up, block more stuff get into the small intestine when we've already gotten some food, and the small intestine's trying to deal with that. Yeah, Kyle?
I have two questions. So
Yeah.
sphincter body.
Yeah.
Going to file with the duct there, but
Yeah.
it also allow the pancreatic juice to go.
Absolutely, it is both. Yeah, it is both. Yeah, there's kind of two separate ones, but they're kind of collectively known as that one sphincter, and they're typically always controlled together too. So yeah.
My second question, could you argue that somatostatin could also be like pelvic sphincter relaxation since it is relaxing the pelvic sphincter?
Yeah, for sure. Yeah, that is another one. Did I forget to write it down or just not have it in my notes? Yeah, because that was, yeah, that's on the previous, I didn't have it here probably, but it was on the video one. Yeah, I just forgot to write that one down. So yeah, the relaxation would be another one. Yeah, the GHIH there. All right. Yeah, thank you. Like your level of detail, Kyle. Good job. Yeah, Ava.
Correct, right, for PHIH is both in the stomach and small intestine for that first point.
Say that one more time, the last part. How it's both what? Mm. Oh yeah. Yeah, so it is really just again, this is our diffuse endocrine system, right? So it's just individual cells scattered throughout the lining of the different parts of the GI tract. And so if we have the cells there that do that, then they'll do the releasing there. So it just happens with that one that we have cells kind of scattered throughout different parts of the GI tract that specifically make somatostatin and release it in those spots. So yeah. Yeah, go ahead.
For the inhibiting secretions, is that SS somatostatin for the gastric and small intestine?
Yes, somatostatin. Yeah, sorry about that one. Yeah, yeah, Chisholm. Yeah. Motilin. Yeah, where did I put that? Okay. What were the two? So I said presence of fats and acids. And I said, yeah, we'll talk about that next week, a little bit more detail. So there's a couple different stimuli for that one. But yeah, for this here, this is fine when we talk about what are called the mass action contractions of the intestines. We'll get to that next week. That's where motilin kind of comes in, and that's more like the fasted state part of it. But it really kind of works in both of those different areas. So it works when there's food there to push it through, but then it also works, yeah, to kind of keep things moving through especially through the lower intestines. So we're kind of pushing everything into the large intestine and not letting bacteria and things travel back to the small intestine. So kind of two separate applications of it, but it does work with both. So thank you. Yeah, good eye. Other questions on that? Okay. All right. Yeah, let's see. I think, oh yeah, we got to do this for sure. We got to do the scorecard here for sure. So yeah, we've got a couple different things here. So we've talked about all the stuff that's dumped into the GI tract, right? But most of this we bring back. Okay. So the total input, if you added all that up, gets you to about 9 liters per day. Okay. So we can see that. So we had like 1,500 in the salivary glands, stomach 2,500, 500, half a liter from the bile, 1,500 from the pancreas, another liter from the intestine, right? So that's about 7,000 or 7 liters, right? So those are all the endogenous secretions, right? Typical just fluid intake might be about 2 liters, right? So that's 9 liters of fluid that goes into our GI tract, right? How much fluid do we have in our blood? About 6, right? So our full blood volume is only about 6 liters, right? So we dump in over our full blood volume. It's not blood, obviously, but over the equivalent of our full blood volume into our GI tract every day, right? So most of that gets reabsorbed, obviously, or we'd be in big trouble, right? And this is why chronic diarrhea is life-threatening, right? If we lose all that, we're not going to last long, right? So most of that comes back in the jejunum, some in the ileum, right? That's all small intestine, right? And then we eventually get all of that back. So by the time it's actually in our stool, it's only about 200 mils, right? So water is just kind of the medium for everything that's put into the stomach and small intestine all the way through. But then we got to get all that water back or we're going to be in trouble, right? We can't lose all that fluid. So, yeah, so most of it is most of that water reabsorption is sodium dependent, okay? And then there's lots of different ways that we bring sodium back, right? Secondary active transport with glucose. We'll talk about that one tomorrow. Exchange with hydrogen coupled with chloride, epithelial sodium channels as well. Okay, so all of these are helping us pull sodium back, and then water follows that sodium for the most part, right? So that's how we're reabsorbing most of that water is along with sodium too. So, okay. Questions about that? All right. Let's actually just talk about diarrhea and constipation real quick. Well, no, we'll do that tomorrow. It's only got a minute left. We won't rush that. Okay. All right. We'll continue on tomorrow, guys. See you in the, yeah, Haley, go ahead. No, I don't have them all graded. I just looked at the ones that might have not gotten under or not gotten above 70% based on the multiple choice only, you know, and then just graded those just so we can get the remediation process going, but I have about half of them done. So, yeah.
That'd be out 5.
I set the email to go at 10 a.m. So in 1 minute. Yeah. Yeah. So.
You're like, here's the email.
Yeah.